So we're on to the continuation of the former lecture on large-scale production of proteins from recombinant microorganisms. Now this is a figure of the replenishment of the TCA cycle in E. coli by the introduction of gene from R. etley encoding pyruvate carboxylase. This avoids the conversion of pyruvate to acetate. The TCA cycle may also be replenished by the introduction of a gene encoding aspartase, converting aspartate in the medium of fumarate. Note that the conversion of glucose to a biomass is a multi-step process, coenzyme A, or CoA. Now, in continuation with the subject matter proper, aspartase, under the control of the strong type promoter on a stable low copy number plasmid. Aspartase activity is induced by the addition of IPTG at the mid to late log phase of growth. The target recombinant protein is introduced on a separate plasmid using the system in minimal medium containing aspartate. The production of different recombinant proteins could be increased up to 5-fold with 30 to 40 degree more biomass production. Bioreactors. A cursory examination of the biochemical engineering literature may give the impression that there is a limitless number of bioreactor designs. However, closer inspection reveals that virtually all of these designs fall into three fundamental classes: stirred tank reactors, STRs, and which may have internal mechanical agitation; bubble columns, which rely on the introduction of air or another gas, spargin for agitation; airlift reactors, which which have either an internal or an external loop. The mixing and circulation of the culture fluid in these reactors are the result of the motion of an introduced gas, usually air, right, which causes density differences, all right, within the different parts of the bioreactor. The traditional and by far the most commonly used bioreactor is the STR. This type of bioreactor has several advantages over other bioreactor configurations. It has highly flexible operating conditions. It is readily available commercially. It provides efficient gas transfer to the growing microbial cells, or in the words of fermentation engineers, the volumetric mass transfer coefficient, KLA, of STRs is high. It has been used extensively by fermentation engineers and microbiologists for growing a variety of microorganisms. In an STR, gas, usually air, is added to the culture medium under pressure through a device called a sparger, which can be either a ring with many small holes or a tube with a single orifice. All the sparging rings generate smaller bubbles and consequently create better initial gas distribution. Sparging tubes are often preferred in many small-scale applications, less than 20 liters, because they are less likely to become plugged. Excuse me. And so thorough dispersion of the gas within the bioreactor requires one or more impellers agitators in addition to the sparger. Mechanical agitation of the culture medium by the impeller breaks down larger bubbles into smaller ones, disperses the bubbles throughout the medium, and enhances the residence time of the bubbles in the bioreactor. At higher levels of agitation, the mean size of the bubbles in larger bioreactors is essentially independent of the size of the holes in the sparger, the type of impeller, its rotational speed, and the physicochemical properties of the liquid phase are important factors that give rise to efficient gas dispersion. In large bioreactors, however, if the initial gas distribution from the sparger is not uniform across the tank, even vigorous agitation may not cause a homogeneous gas environment. Because of the corrosive or abrasive nature of many culture media and sterilization procedures, STRs are usually constructed from stainless steel or glass. The glass units are usually limited to laboratory scale bioreactors that have a capacity of less than 50 liters. One limitation on the size of a bioreactor is the ability of the system to efficiently remove heat that is generated as a consequence of either the metabolism of the growing cells or the energy input by agitation. Too much heat raises the temperature and alters the physiological state of the cells and decreases the product yield. Heat can be removed by using a coolant jacket around the reaction vessel or by internal coils. Although internal coils are more effective than jackets in keeping the fermentation reaction close to the desired temperature, they can become coated with microorganisms, which prevents cooling, and they sometimes interfere with the proper agitation of the fermentation broth. Contamination of a fermentation with fungi or bacteria is usually disastrous. Therefore, bioreactors are designed so that they can be sterilized, usually with pressurized steam. There should be no internal dead spaces or surfaces that escape contact with the steam during sterilization. All seals, probes, and valves also must be readily steam sterilizable. When a bioreactor is designed, a trade-off is often made between a full set of ports for probes, which enables monitoring of the fermentation parameters, and fewer ports, which makes the maintenance of sterility easier. The high level of agitation of the culture medium during a fermentation reaction often causes considerable foaming. Excessive foam can wet the filter at the port through which the introduction or the introduced air exits the bioreactor, thereby both decreasing the airflow and providing a potential pathway for the entry of contaminating cells. Either chemical antifoaming agents or mechanical foam breakers can be used to control foaming. However, the chemical agents can diminish the content of microbial growth by preventing oxygen transfer, or in some cases by inhibiting cellular enzymes. Furthermore, if an antifoaming compound is not removed before downstream processing, it can contaminate the final product. Foaming can also be controlled by providing sufficient headspace within the bioreactor, that is, space above the liquid, in which the bubbles can dissipate. In practice, then, the working volume or actual volume of the culture in an STR is typically only about 75% of the total volume of the bioreactor. Many of the considerations that apply to STRs also apply to pneumatic reactors, such as bubble columns and airlift bioreactors. Thus, for example, sterility, constant pH, and constant temperature are key components of any fermentation, regardless of the precise configuration of the bioreactors. The configuration of bubble columns and airlift bioreactors give them some distinct advantages over STRs. These pneumatic reactors are more energy efficient than STRs because agitation is provided by the injection of a stream of air or another gas if anaerobic microorganisms are being grown rather than by a mechanical stirrer. Also, with the elimination of the mixer shaft in these units, there is one less potential site of entry for contaminating organisms. Pneumatic reactors generate a lower shear environment than do STRs. Also, in airlift reactors, the shear stress is more evenly distributed throughout the vessel than STRs. The reduction of shear forces is important for the following reasons. Genetically engineered microorganisms are more often susceptible to lysis when exposed to shear stress than are unmodified organisms, because the extra metabolic burden of synthesizing a foreign protein often causes genetically engineered microorganisms to form weakened cell walls. A frequent cellular response to the shear forces is decreased synthesis of all cellular proteins, including the recombinant protein. Shear stress can alter the physical and chemical properties of the cell so that the downstream processing steps become more difficult to perform. For example, the fermentation conditions can inadvertently increase the amount of surface polysaccharides that a microorganism produces, and as a consequence can change the conditions for effective harvesting and lysis of these cells, making it more difficult to purify the target protein. In bubble columns, the air is introduced under high pressure near the bottom. but the smaller bubbles coalesce into larger ones as they rise through the column, leading to an uneven gas distribution. In addition, the use of high-pressure air tends to cause excessive foaming of the medium. These disadvantages restrict the flexibility or effective range of operating conditions as well as the potential size of bubble columns. Airlift bioreactors, however, can be readily adapted for either pilot plant or large-scale fermentation process. In an airlift reactor, the gas is introduced into the form of a vertical channel riser. Both the gas and liquid follow up the rise until they reach an open space at the top, gas-liquid separator, where the gas is at least partially disengaged from the liquid. Excuse me. The degas liquid is which is denser when the than the gas liquid. This ends in a separate vertical channel down, and moves along the base of the reactor until it reaches the bottom of the riser. In this way, the culture fluid and cells are continuously being circulated around the bioreactor. There are two main types of airlift bioreactors. Those that have a single container with internal baffles that create interior liquid car circulation channels, internal loop reactors, and those that have an external loop so that the culture liquid circulates through separate independent channels, external loop reactors, as seen in 17.9D. Internal loop aircraft reactors are simple in design, but once they are constructed, both the volume and circulation are fixed for all fermentation processes. In contrast, the external loop airlift reactors can be easily changed or modified, example by altering its volume to suit the requirements of different fermentations. Airlift bioreactors are generally more efficient than bubble columns, especially for denser or more viscous suspensions of microorganisms. In airlift reactors, mixing is generally better. The bubble coalescence is not as big a problem as it is in bubble columns. In extremely large airlift fermenters, such as a 1,500,000 liter fermenter built by Imperial Chemical Industries Limited, England, for the production of single-cell proteins. It takes a considerable amount of time for a cell to complete a full cycle through the reactor to prevent the cells from becoming substrate depleted while they are traversing the bioreactor. There are multiple injection points for the introduction of substrate along with the length of the unit. Typical large-scale fermentation systems. When recombinant microorganisms are used to overproduce products such as pharmaceuticals like insulin, cells are typically grown to mid to late log phase of the growth cycle so that the target protein levels be optimal. On the other hand, when recombinant microorganisms are used as factories to synthesize useful metabolites, such as antibiotics, host cells are commonly grown in the deceleration of stationary phase, where the synthesis of secondary metabolites is often optimal. Clearly, these sorts of differences must be considered when large-scale fermentation process is being developed. For maximum protein production, it is generally preferable to use clone genes that are under the control of strong promoters that can be regulated. Initially, it was thought that constitutive expression of a clone gene would be sufficient to obtain reasonable quantities of the product. However, experience has shown that continuous transcription and translation of a cloned gene drains energy from essential cell functions and slows cell growth. Excuse me. Cloned gene drains energy from essential cell functions and slows cell growth. With an inducible system, the expression of a cloned gene can be confined to a specific period in the growth cycle of the microorganism. On these grounds, for optimal protein production, the process should be consisting of two relatively high cell density. Second, depending on the nature of the promoter, that drives the clone gene, drives the clone gene. Transcription is induced either by shifting the temperature or by adding a chemical inducer such as IPTG to the medium. A two-stage system is not easy to implement in a large bioreactor greater than 100 liters because it is technically difficult to raise the temperature quickly from 30 to 40 degrees centigrade or to ensure that this chemical inducer is rapidly and evenly mixed in a large vessel. Moreover, many chemical inducers such as IPTG are too expensive to be used on a large scale. However, as discussed earlier, this problem can be overcome by using the two connected bioreactors, two-stage fermentation, so the cells are grown in these conditions in the first vessel and the induction is carried out in the second. Under these conditions, growth and induction are separately optimized, thereby increasing the overall amount of product formed per unit time productivity of the fermentation. The next would be two-stage fermentation in tandem airlift reactors. Let's see what happens. Two-stage fermentation in tandem airlift reactors. E. coli NM989, which carries the gene encoding the enzyme T4-DNA ligase under the transcriptional control of the FPL promoter and a temperature-sensitive CI repressor, was grown and induced in a two-stage airlift bioreactor. In this bacterial strain, the T4-DNA ligase gene was integrated in the chromosomal DNA, a location obviating any potential problems or plasmid instability that might occur during extended fermentation. The growth stage was carried out at 30 degrees centigrade in an external loop airlift bioreactor that had a 10-liter working volume. The T4-DNA ligase gene was not expressed under these conditions. A second external loop airlift bioreactor with a working volume of approximately 5 liters at 42 degrees centigrade was used for the induction stage. The two bioreactors were linked by a transfer tube with a pump that controlled the continuous flow of cell suspension from the growth stage bioreactor first into the induction stage vessel second. In addition, cells suspended in culture medium were removed from the induction stage bioreactor at a specific rate and prepared for downstream processing. The maximal specific growth rate Mumax of the microbial culture was approximately 0.66 reciprocal hour in the first bioreactor and 0.54 reciprocal hour in the second. These values correspond to cell doubling times of 63 and 77 minutes, respectively. Fresh medium is continuously added at a rate of 2 liters per hour to the growth stage medium, growth stage ferment, and cell suspension effluent was simultaneously removed from the induction stage fermenter at the same rate. As a consequence of the liquid volumes of the two bioreactors, on average, cells spend about 5 hours in the growth stage bioreactor and 2 hours in the induction stage bioreactor. The different residence times in the two phases of this fermentation process were necessary to optimize the number of cells produced and the yield and stability of the T4-DNA ligase. Generally, residence times can be altered as required by adjusting the relative working volumes in the two fermenters of a two-stage system and by adjusting the volumetric rate of input of nutrients into the first bioreactor. The double external loop design of the airlift fermenter used in this work facilitated the adjustment of the working volumes of the two fermenters relative to one another. It is also added versatility to the system so that it was possible to obtain a variety of different growth conditions for different populations of recombinant cells. For T4-DNA ligase production, the best results were obtained when approximately 33 milliliters of cell suspension was transferred every minute from the growth stage bioreactor to the induction stage bioreactor. Because this amount of cell suspension was equivalent to only 0.67% of the volume of the induction stage bioreactor, the incoming cells underwent a virtually instantaneous temperature shift from 30 to 42 degrees centigrade. Nutrients in a bioconcentrated form were added to at a specific rate at the induction stage bioreactor throughout the fermentation process to keep the cells in this bioreactor in log phase. This action prevented the T4-DNA ligase from being degraded by the proteolytic enzymes that are normally synthesized during the deceleration and stationary processes. With the continuous two-stage bioreactor, induced E. coli NM989 can be grown to a density of approximately 4 grams dry weight of cells per liter of culture after induction. About 4% of the cell protein is T4 DNA ligase, an amount that corresponds to approximately 25,000 units of enzyme activity per gram dry weight. This process can produce approximately 100,000 units of enzyme activity per liter of culture, or about 4.8 million units per day, assuming that it is possible to recover about 20% of the initial activity following purification of the enzyme, and that the enzyme sells for about $0.25 per unit, $0.25 per unit, that the final yield of purified enzyme in one day will be worth about $240,000. Although these calculations do not consider all of the costs that go into the production of protein from a genetically engineered microorganism, it is clear that for highly valued products, small to moderate sized continuous fermentation systems can generate significant returns on the initial capital investment. Two-stage fermentation in a single-stirred tank reactor. The tripartite fusion protein Ag-beta-gal, which is used for immunological assays, was produced on a large scale in a single STR. The gene encoding Ag-beta-gal was constructed by recombinant DNA techniques and encodes the 5 immunoglobulin G-binding regions of Staphylococcus aureus, protein A, 2 immunoglobulin G-binding regions from Streptococcus sp strain, G148 protein G, and beta-galactosidase from E. coli. The synthetic Ag-beta-galactose gene was placed under the control of the bacteriophage lambda PR promoter, which is regulated in the same manner as a peer promoter, cloned into a plasmid that carries the gene for ampicillin resistance and introduced by transformation into E. coli. The strain with the Ag-beta-gal plasmid carries a second plasmid that has the genes for a temperature-sensitive CR repressive protein and a kanamycin resistance gene. A 5-liter volume of these cells was grown in an STR at 30 degrees centigrade in the presence of both ampicillin and kanamycin to provide selective pressure for the retention of both plasmids, and then used to initiate growth without antibiotics at 30 degrees centigrade in a 45-liter STR. The cell suspension in the 45-liter fermenter in turn served as the inoculum for a 600-liter STR, where the cells were grown at 30 degrees centigrade without antibiotics. In general, to keep the cost of the process to a minimum, antibiotics are not needed to large-scale microbial cultures. When the cell density in the 600-liter bioreactor reached the equivalent of about 4 grams dry weight per liter of growth medium, the temperature was shifted from 30 to 40 degrees centigrade to induce expression of Ag-beta-gal protein. Under these conditions, it takes about 1 hour to reach 40 degrees centigrade. A temperature of 40 degrees centigrade rather than 42 degrees centigrade was chosen because the lower temperature was found to yield the same level of Ag-beta-gal protein while allowing the cells to grow for a longer time. In other words, the lower temperature 40 degrees centigrade conditions yielded a larger amount protein product. The specific activity of the Ag-beta-gal protein increased for 2 hours after the initiation of the induction. Of the Ag-beta-gal protein increased 2 hours after the initiation of the induction and then decreased. This decrease in the activity was probably due to the synthesis of proteases by cells that entered the deceleration and stationary phases of the growth cycle. In addition, 50% of the cells had lost their plasmids after growth for 4 hours at 40 degrees centigrade. These problems were notwithstanding after 4 hours at 40 degrees centigrade. The Ag-beta-gal protein was approximately 20% of the dry weight of the total biomass. Considering the very high yield of the target protein that is produced by this strategy, it is probably not necessary to integrate the genes for the Ag-beta-gal protein and the CR repressor into the chromosomal DNA of the E. coli host cell in an effort to increase the final yield. batch versus fed batch fermentation. In some instances, a simple fed batch strategy can be used to produce both a high cell density and a high level of expression of a target protein. For example, a plasmid carrying a gene encoding a hybrid protein that includes an insulin B peptide under the control of the E. coli tri-promoter was introduced into a TRP minus mutant strain of E. coli that cannot be synthesized to tryptophan, and the transformant was grown in media containing various amounts of tryptophan. At high levels of tryptophan, the synthesis of the target protein was repressed. However, after consumption of the tryptophan in the medium by the growing cells, synthesis of the target protein was induced. With this system, the addition of tryptophan to the medium resulted in batch cultures and increases in the amounts of both the biomass and target protein produced. However, fed batch fermentation was more effective than batch fermentation with or without added tryptophan. However, after consumption of tryptophan in the medium by growing cells, synthesis of the target protein was induced. With the system, the addition of tryptophan to the medium resulted in batch cultures and increases in the amount of biomass and the target protein produced. However, fed batch fermentation was more effective than batch fermentation with or without added tryptophan. In another experiment, interferon gamma was produced in E. coli. Expression of the interferon gamma gene was controlled by the PL promoter regulated by the temperature. Sensitive Cl repressor. Cells that contained this construct on a plasmid were grown in either batch or fed batch mode with both stepwise and constant rate medium feeding strategies. In the fed batch mode, the addition of growth medium was carried out simultaneously with the temperature induction of the PL promoter in the late exponential growth phase. Use of the fed batch mode resulted in a significant increase in the length of the cell growth phase following induction. This fed batch strategy enabled researchers studying the system to achieve a cell biomass that was 5-fold higher and a final IFN interferon gamma concentration that was 23-fold higher than they were able to achieve in batch culture. Another group studied the fermentation conditions that yielded the optimum expression of a monoclonal antibody Fab fragment directed against tetanus toxoid under the transcription control of the E. coli lac promoter. The plasmid construct included signal sequences that were inserted immediately upstream of the antibody light and heavy genes to target the antibody fragments to the E. coli periplasm. In this case, inexpensive lactose rather than the considerably more expensive IPTG could be used to induce the expression of the monoclonal antibody fragment gene, provided that the host strain of E. coli was lac and therefore able to take up lactose from the medium and convert it to glucose and galactose. In fact, allolactose, an isomer of D-lactose and not D-lactose itself, is the actual inducer of the E. coli lac operon and is formed only if the host strain of E. coli contains a small amount of beta-galactosidase expressed see chapter 6. An important facet of these experiments is that lactose not only acts as an inducer but also is metabolized, providing an additional carbon source for the Fab-producing E. coli cells, thereby supporting both cell growth and production accumulation. Again, in these experiments, lead batch fermentation was clearly superior to batch fermentation, yielding both a larger amount of cell biomass and a greater concentration of the Fab fragment. Fed batch fermentation strategies have also been successfully employed in the production of non-protein products such as poly-3-hydroxybutyrate and poly-3-hydroxybutyrate co-3-hydroxyvalerate biopolymers with plastic-like properties. In this case, E. coli host cells carried a plasmid that contained polyhydroxyalkanoate biosynthesis genes from the bacterium alkali genes lattice by using an inexpensive growth medium such as whey, a waste byproduct of cheese making that consists mainly of lactose, to produce these polymers. It is hoped that a commercially viable product can be produced on a large scale. Another group reported that instead of a single introduction of lactose in a fed batch fermentation to produce active protein fragments of human apolipoprotein A, a strategy that employed continuous induction of the lactose beneficially influenced the expression of the target protein with a 75-liter fermenter using lactose as a sole feed was not efficient for cell growth, presumably because the host strain of E. coli was unable to metabolize galactose. However, with a 1:50 ratio of galactose to glycerol, the target protein reached 16% of the total cellular protein. It remains to be seen whether continuous lactose induction will benefit the expression of proteins other than human apolipoprotein A, and to what extent this process can be scaled up. Harvest microbial cells. The first step in the process of purification of a product that is synthesized during microbial fermentation is the separation of the cells from the culture medium. Recombinant and native microbial cells can both be harvested with the same type of equipment. However, as a consequence of physiological changes such as alterations in cell size and the production of extracellular polysaccharides, conditions that have been established for non-transformed cells may not be optimal for recombinant cells expressing a foreign protein. For large volumes, either high-speed centrifugation, which is the current method of choice, or membrane and microfiltration is used to separate cells from the growth medium. High-speed semi-continuous centrifuges have been specifically designed for the harvesting of microbial cells. The cell suspension is continuously fed into a running centrifuge, and the cells are concentrated within it, while the clarified medium is collected in an external container. When the centrifuge chamber is all full of packed cells, the run is stopped and the cells are removed. The need to stop and then restart the procedure periodically, especially when large volumes are being processed, can be a major inconvenience. Furthermore, the cost of both the equipment and the power to run it, the release of the microorganisms into the air as aerosols during the harvesting procedure, and the difficulty in removing all the microbial cells from the spent medium is also limit the use of this separation medium procedure. Membrane filtration is an alternative method of separating the cells from the culture medium. Unfortunately, with traditional dead-end filtration, the microbial cells accumulate on the surface of the polymeric membrane. Membrane filter. Consequently, the flow rate of spent medium through the membrane decreases rapidly. Increasing the pressure on the membrane enhances the flow for a short time. However, the cells still accumulate on the surface of the membrane and may even form a more compact and less permeable layer as a result of the pressure. An alternative filtration technique entails passing the cell suspension at a high speed across the surface of the membrane for filtration. Under these conditions, only a very small fraction of the circulating liquid actually goes through the membrane in any one pass. The remaining cell suspension acts to sweep the membrane clean of accumulated cells so that the rate of liquid flow through the membrane does not decrease as rapidly as it does in dead-end filtration. In both deadened and cross-flow filtration, the average pore size of the membrane is 0.2 to 0.45 micro-m. After many cycles in a cross-flow filtration system, almost all of the culture medium will have passed through the membrane. The use of let's just continue there. The use of cross-flow filtration is generally limited to laboratory scale operations. More industrial scale operations still rely on centrifugation. The next step in the purification process depends on both the nature and location of the product. If the final product is a protein and it is in the culture medium, the medium is concentrated often by ultrafiltration, and the target protein is purified by column chromatography or other standard procedures. If the product is a low molecular weight compound in the culture medium, it can be purified by the appropriate extraction procedures. Finally, if the product is in the cellular fraction, the cells must be disrupted, lysed, before the steps leading to production purification are initiated. Disrupting microbial cells. A large number of chemical, biological, and physical methods have been developed for disrupting microbial cells. All of these procedures represent a compromise because they must be vigorous enough to break the microbial cell walls, yet gentle enough to ensure that the protein product is not denatured. There is no single set of conditions for cell wall lysis because the cell walls of diverse microbial species are composed of different polymers. In gram-negative bacteria, the cell wall is external to the cytoplasmic membrane and consists of a thick peptidoglycan layer of acetylglycosamine and N-acetylmuraminic acid residue cross-linked by oligopeptides. The cell wall of gram-negative bacteria has an outer membrane, a thin peptidoglycan layer, and the cytoplasmic membrane. The yeast cell wall is composed of a thick layer of partially phosphorylated mannans and beta-glucan. Cell wall composition. And strength depend on culture conditions, the cellular growth rate, the phase of the growth cycle, and when the cells are harvested, how the concentrated cells are stored, and whether the isolated microorganisms was expressing a clone gene. All these factors affected the cell's susceptibility to disrupt. The chemical methods to disrupt microbial cell walls include treatment with alkali, organic solvents, or detergents. If the protein product is stable at pH values from about 10.5 to 12.5, the bacterial cell lysis can easily be carried out on a large scale at low cost. For example, recombinant human growth hormone is efficiently released from E. coli by treatment with sodium hydroxide at pH 11. Few, if any, viable cells remain after alkali treatment, which obviates concerns about the inadvertent release of a genetically engineered microorganism from a production facility. Treatment with an organic solvent is a simple and inexpensive way to disrupt cells and has been used for the isolation of enzymes from yeast. However, preliminary tests must be run to make sure that the proposed treatment dose treatment does not denature the target protein. Detergents permeabilize bacterial cells by solubilizing cell membrane and membrane proteins. As a consequence of this activity, holes are formed and proteins and other molecules are released from the cells. Unfortunately, detergents are expensive, frequently denature the protein product, and often retained as contaminants throughout the purification process. The major biological methods for disrupting microbial cells is enzymatic lysis. For example, the cell walls of gram-negative positive bacteria are readily hydrolyzed by the enzyme lysozyme, which is isolated from egg whites. The cell walls of gram-negative bacteria are hydrolyzed by lysozyme, and the cell walls of yeast are hydrolyzed by combinations of one or more of these following enzymes: beta-1,3-glucanase, beta-1,6-glucanase, mannanase, and chitinase. Enzymatic treatments are highly specific, and the conditions for lysis are mild. Currently, cost considerations limit the use of enzymes as cell lysis genes. However, the use of genetically engineered microorganisms for large-scale production of the enzymes that attack cell walls should make them less expensive. Microbial cells can be physically disrupted either by non-mechanical methods, which includes osmotic shock, a repeated cycle of freezing and thawing, or by mechanical procedures such as sonication, wet milling, high-pressure homogenization, impingement. Generally, after treatment by a non-mechanical method, many of the cells remain intact. In contrast, mechanical disruption is highly efficient, which makes it a preferred choice. A sonicator that generates high-pressure sound waves that cause cell disruption by shear and cavitation production of internal holes is generally useful for small volumes. Wet milling is quite commonly used for disruptive large quantities of cells. A concentrated cell suspension is pumped into the chamber of a high-speed agitator bead mill that is filtered with an inert abrasive material. Such as small glass beads less than 1 millimeter in diameter and is fitted with a central shaft that has a number of attached blades. When the device is turned on and the blades are put in motion, most of the cell's disruption occurs as a consequence of the shear forces generated by the high-speed motion of the glass beads. Optimized cell division depends on both the number of the configuration of the agitator discs, the agitator speed, the size of the glass beads, the number of glass beads, the cell concentration of the geometry of the grinding complex, the grinding chamber, and the temperature of bead mills have been successfully used to disrupt a wide range of different kinds of microbial cells and can readily break recombinant as well as non-recombinant cells. In the high temperature, high pressure homogenization process, concentrated cells are pumped into a valve assembly under high pressure, and the pressure is then rapidly decreased, causing the cells to lyse. This process can be customized for different microorganisms and protein products by changing the operating pressure, the design of the valve, the temperature of the cell suspension, or the number of times the cell mass is treated. The forces that are created at the points of contact disrupt the cells with a device called a microfluidizer. For example, two parallel streams of equalized cells in suspension are directed toward one another. With this device, a high percentage of cells are disrupted by a single passage through the unit. However, additional passages may be required for complex breakage of other cell types. Unlike high-pressure homogenizers and high-speed agitated bead mills, which generally require highly concentrated cell suspension, this device can be used to disrupt cells in either dilute or concentrated preparations. The activities of cellular proteins are not significantly impaired by the technique. When the cell suspension is treated with low levels of lysozyme and then disrupted in the microfluidizer at a much lower level than normal pressure, the fluid and fluid velocity, the activities of labile proteins, which might otherwise be inactivated by high temperature, are retained. The downstream processing. After cell disruption, cell debris is removed by either low-speed, high-capacity centrifugation or cell membrane macrofiltration. The protein product is precipitated from the crude lysate, the clarified lysate, or the cell-free culture medium with organic solvents, alcohol or acetone or ammonium sulfate. Under these conditions, the target protein is usually enriched approximately 2 to 5-fold. Unfortunately, the cost of the Unfortunately, the cost of the precipitant can add significantly to the cost of the process. Alternatively, the crude oil protein mixtures are concentrated and fractionated by cross-flow ultrafiltration through membranes that have a smaller average pore size than those for the either cell concentration debris or debris remover. This approach can be used with volumes ranging from 1 liter to several thousand liters and can be performed continuously, which means that large volume systems are not required depending on the size and properties of the target protein. This method can yield 10 to 100-fold enrichment. On a large scale, it is impractical to remove all small molecules such as salts or organic solvents from protein solutions by convert to conventional lab procedures. Consequently, the same apparatus that is used to concentrate proteins by ultrafiltration has been developed for the large-scale dialysis proteins. In addition, by using two different types, thumb-sized membranes, large volumes of target proteins with different molecular masses have been fractionated by ultrafiltration. The required degree of purity of the final protein product depends on its end use. In some cases, such as in enzymes for using the laundry detergent, crude preparations are satisfactory, but for other products, such as pharmaceutical proteins, additional purification procedures are required. A number of proteins that are overproduced intracellularly are confined to insoluble particles, inclusion bodies within the bacterium. After disruption, such inclusion bodies can be subtly separated from the bulk of the remaining cell components. Initially, research has found it difficult to solubilize inclusion bodies without irreversibly denaturing the proteins. But protocols have now been devised to renature the protein found in the inclusion bodies. Of course, the additional steps increase the cost of the purification process. And now the next topic would be protein solubilization. In some instances, overexpression of a target protein can result in the production of both soluble and insoluble forms, which complicates the production and purification process. For example, when human insulin-like growth factor IgFI, a 7.6 kilodalton SCDA peptide, was expressed in E. coli cells, approximately 90% of the recombinant polyprotein was localized in the E. coli medium, or the E. coli periplasm, soluble and insoluble, and about 10% is found in the external medium soluble. To recover both the forms of IgFI from the periplasm, high concentrations of urea and diethiotriator at alkaline pH were added to solubilize insoluble forms of the peptide in situ. This treatment kills but does not lyse the cells. The solubilization procedure produces a highly viscous solution which precludes removing the cells and all cell debris by centrifugation. Instead, an aqueous two-phase liquid extraction procedure that separates both soluble and soluble materials were developed for this purpose. Now, both here in situ the solubilization and aqueous two-phase liquid extraction procedure are highly efficient. 80 to 95% of the IGFI is discovered from 10 to 100 or 1,000 liter fermentation of these methods. What's the next topic? Large-scale production of pure plasmid DNA. Gene therapy and genetic immunization protocols are being tested in a large number of clinical trials. Many of these trials require plasmid as vectors to deliver the remedial DNA to the patient. As these procedures become more routine and extended to many more patients, it will be necessary to produce plasmid DNA initially in the 5 to 20 kilo base size range on a large scale and in a highly purified form that is the pharmaceutical grade. A number of factors, including the choice of a host strain such as E. coli K12 that is safe and well characterized, growth oxidations, defined growth conditions, defined medium, and the purification process that removes all of the undesired genomic bacteria. Undesired genomic DNA, RNA proteins, lipids, and liposaccharides need to be considered for the large-scale production of plasmid DNA. Generally, to ensure that the plasmid DNA will be stable, a host strain should produce only 2 levels of nucleases. In addition, the presence of high antibiotics in the culture medium should be avoided since it is difficult to remove all traces of an antibiotic from the final preparation from DNA isolation. The current method of choice for cell breakage is the alkaline lysis procedure. Treatment with 0.2 NaOH and 1% sodium diodecyl sulfate, which breaks cells without disrupting plasmid DNA. The lysis solution must be added so that mixing is sufficient to lyse all the cells and local pH extremes and avoided by the shear that is generated by the mixing does not damage the plasmid DNA. After cell lysis, the precipitates formed, which contain cell debris, denatured proteins, and nucleic acids must be removed. This is most commonly done by centrifugation using fixed angle rotors, a process which is difficult to scale up. After removal of the precipitated material, plasmid purification is best achieved by anion exchange chromatography, followed by size exclusion, gel filtration, chromatography in the industrial production of many recombinant proteins. High-pressure homogenizers are used to continuously disrupt cells. However, the use of such homogenizers typically results in the degradation of a large portion of the plasmid DNA as a consequence of the severe shear stress generated during cell breakage. And to work out a process for the large-scale preparation of a plasmid DNA, one group of researchers developed and optimized a unique continuous thermal lysis protocol. In this procedure, cells were harvested after high cell density batch culture, filtered to remove most of the growth medium, and resuspended in lysis buffer containing lysozyme. After incubation at 37 degrees centigrade and gentle stirring for 20 minutes, these cells were heated to 70 degrees centigrade for 20 seconds and then filtered to remove all the cell debris. Now, this lecture should be over in a minute from now, and the rest of it, if anything, will be continued in the next lecture, right? And after incubation, 37 degrees centigrade and gently stirring for 20 minutes, the cells were heated to 70 degrees centigrade for 20 seconds and then filtered to remove the cell debris. Using this procedure, researchers have reported obtaining 100 mg of high quality, 100 mg, 100% of high cell density cell culture. Plasmid DNA free of contaminating chromosomal DNA per liter of high cell density culture. Use of lysozyme is the most expensive component of the process. As a consequence of the high plasmid yield, the avoidance of time-consuming expensive centrifugation steps and short processing time, 17 liters of E. coli cells can be processed in 45 minutes and the ease with which process can be scaled up, this approach is likely to provide a highly effective means preparing large amounts of highly purified plasmid DNA. So this lecture is just about over, and the summary and the illustrations will be explained in the next lecture, right? So make notes while I come up with the preparation of the next. Take notes and do enough research, and best wishes, right? Thank you.